Good morning, everyone. Welcome to Lecture 6. Before we get to Lecture 6 on the gene regulation, I'd like to give a very quick review on the mechanism of gene expression, especially the transcription and the translation, because that forms the foundation on how those processes can be regulated. And so we know when gene expression, when you need to make a protein, the genetic information needs to be used to start a process of transcription to make mRNA, and then the mRNA will be translated to protein. And last week we discussed in detail about how this transcription can be is started with these two essential elements, right? The RNA polymerase and sigma factor. And then once it started, the sigma factor goes away, the RNA polymerase continues, the new mRNA is made, eventually released. Now, in this particular process, if we look at the start of the transcription, and this is where the complexity comes in. So the transcription initiation, or the start of the transcription, is determined by the sigma factor. And as we talked about, sigma factor has two binding sites, and so these are the two binding sites. One is what's called the Tata box, or -10 region, or it's called the Prinal box. I'm sorry, you have three different names to worry about. They're the same thing that could be called. And then the second binding site is -35 region. And what's listed here, just to emphasize how important are these binding sites, or how common, you are presented with 6 different promoters. This region is called promoter. And the 6 promoters all have in common is the two binding sites. And also, it's extremely important, the distance between the two. Okay, that's the distance between the binding sites. And last week, I used the monkey bar and the 2 hand as an analogy, so that I need to hang on to the monkey bar with 2 hands. And now, unlike my hands, the sigma factor is a protein, it's a fixed structure. So its arm cannot move, not like my arm. I can flex a little bit. Okay, and so this is, you need the fact that you need 2 binding sites to have a stable interaction, and then the RNA polymerase can bind and then start the transcription, which means any interference between these binding sites and sigma factor can prevent or weaken or simply shut off the regulation and the interaction. And also, once the binding is going, and this is actually the RNA polymerase is going down the mRNA, the DNA, to make transcription. And so this is a physical process that can also be blocked. And so these are the things that actually being used in regulating how transcription is started. Translation, the start of the translation is requires the formation of the initiation complex. Okay, this is also a complex process, and the translation itself, it cycles, and the more factors involved, you have the ribosome. the large, small unit, and the large unit. And these are all goes in cycles, and which we'll come back to talk about how this could be regulated, right? Now, this is a quick review of the start of the transcription and translation. Does anyone have any questions before I move on? Yes, go ahead.
So the whole point of transcription is to make mRNA in this case?
Yes. Yes, the transcription, the process of transcription is just to make mRNA. So the mRNA then can be translated into peptide. And we explained why this process, and because this then can be the genetic information, the sequence can then be translated into a peptide sequence.
And that's the anti-parallel portion, like the light green portion of the DNA.
Repeat the question again. I'm sorry, I can't.
That's the light green portion of the DNA, or is it the orange one?
The light port, the DNA is a double-stranded, right? And so in this case, they draw to highlight the difference of the two strands. Yes, it's anti-parallel, and you see the the 5' to 3'. Remember the distance? I mean the directions. Yeah, and so and I also highlighted that when when mRNA and DNA forms when they form an anti-parallel structure as well. So in the process of transcription, that helps you understand which strand is being transcribed. All right, which strand is used as a template. In this case, because the mRNA is always made from 5' to 3', right? Because the RNA polymerase and the DNA polymerase can only extend the 3' free hydroxyl group. And and so then based on the anti-parallel, you can then you can know for sure that the template that's being used, which of the strands being used as template is a 3 to 5'. Okay. Any other questions? Okay, I see. You don't need to raise your hand, just jump in, because I can, when you turn on I can actually see you turn on the mic, and so I know there's someone that's want to ask a question, but I can't see your chat because I'm doing the sharing. Okay, so I'm gonna stop the review here and then move on to lecture 6, if there's no additional question. Okay. So in lecture 6, the focus will be regulating gene expression. When we think about regulating gene expression, and the first question is, well, why do you have to regulate gene expression? Wouldn't it just be if you need a protein, You know the protein is needed, just make all of them. Anyone want to chime in why gene expression needs to be regulated?
Because like all the cells have DNA, but they need to know what exactly to produce so that they're not all produce unnecessary things.
Ah, okay. Unnecessary. What do you mean by unnecessary?
Like I saw a video explain like the eyes aren't producing the same like hydrochloric acid in the stomach because that would be very inconvenient. Um, so
Okay. All right. So your analogy is on human body, and you don't produce your stomach acid when there's no food, right? Only when you eat, then you produce stomach acid. It's actually a mechanical mechanism in the stomach. And when you stretch and you get the signal, they start producing acid, stomach acid. Why did I know this? Well, because I was looking at the gut microbes and the digestion. So I looked into this. Normally, this is not something a microbiologist would know. Okay. What about any other reason? So this is for need, and excess acid is not good for you. Any other reason?
Because of the energy, like it uses energy, so like it wouldn't, like, why would it use energy for something that, so the energy is distributed the right way, so using the energy the right way, the more efficient way.
Excellent, excellent. That's precisely the point. Remember, bacteria, we spend a lot of time talking about bacteria living in an energy poor environment, all right? And so they are very, very conscious about conserve energy. So only lately we've learned that when you leave the room, you need to turn off the lights, right? And that's to conserve energy. But bacteria is always need energy. Okay, so we need energy, but can you put some numbers on it? Okay, well, how much energy are we saving here? Are we talking about 1 ATP, 2 ATP? So if we were to talk about a typical bacteria, so I assume you're writing down the notes, right? So if you think about a typical bacteria protein, a typical bacteria gene, so a typical bacteria gene is 1 kb. Let me think about that. Okay. Now, a typical bacteria gene is 1 kb. And then how many, the protein, what about protein? How many amino acids?
You said 3, right? Wasn't it like 3 ATP per amino acid or something per
No, don't jump in too far. That's correct, but let's do a little bit of calculation. So if a typical bacteria gene is about 1 kb, I mean 1,000 base pair long, and that typical bacteria protein gets translated into protein, how many amino acids does this protein have? Right? Okay. So then now you can think about the energy costs. Now to transcribe 1 kb, how many ATP is needed or equivalent is needed? People, anyone want to chime in? To transcribe 1 kb, and how many ATP is needed? And transcribe, you need is one. So you make one mRNA, right? mRNA, and that's how many? How long is the mRNA that's going to be? 2,500.
Professor, can you repeat the process again? So you said that there's 1 kb, which is 1,000 base pairs in a typical bacteria, and then is that like each protein has 333 amino acids?
Yeah, that's for that typical bacteria gene, right? So for a typical bacteria gene, it's 1,000 base pair, because you have to give some numbers, right? So we say a typical bacteria gene is 1,000 base pair long. You can say a typical human is, let's say, 5 foot tall, right? And so that gives you some estimate to do the calculation. So a typical bacteria gene is 1,000 base pair long, and that gene gets translated, transcribed, then translated into a protein. Then that protein would have typically 333, right? Because each amino acid is coded by 3 base pairs, right? Okay, so now now think about the energy cost to transcribe one kb. Then you have to make 1,000 base long, right? So your mRNA is 1,000 bases long, right? And that equals to 1,000 ATPs, right? And then you have to think about translating. And translation. We know translation that produces 333 amino acids, right, for total. And then, right, and then you have to think about energy costs for translating. Yeah, go ahead, Mamadou.
I would like to ask about the 1,000 ATP. How do we get the ATP? Is it the hydrogen bonding? Do we need ATP to do that bonding, or if it is the case, we know that like to bond to like the bonding between oh, I think that one is the DNA. Sorry, because I was talking about like C and G. When they are bonding, we have 3 hydrogen bonds, right? And then A and T, we have 2 hydrogen bonds. So I was just considering that, but that one is for only DNA, right?
No, the answer is what you're describing was not correct, and the energy the hydrogen bond between bases is not that's hydrogen bond is formed by the distance between the two bases. The ATP, the energy is needed to form the phosphodiester bond. That when you think about the DNA polymerase and the RNA polymerase, right? When you extend the DNA, you need the energy to form that bond to extend the ATP, to extend the DNA or extend the RNA. So what I'm detecting is that I would recommend you go back and you read the lecture from last one talking about the DNA polymerase and the RNA polymerase. And that's where the energy is needed. Okay, so we also then talked about translating one amino acid needs two, actually needs three ATPs. Okay, and so altogether, then being this process, what you ended up is that this then requires 2,000 ATPs, right? And so that gives you a reason to say, or justification to say, how expensive is the making the protein, right? Because we know each glucose can only generate 38 ATP at maximum. So you will need a lot of energy, a lot of glucose, burning a lot of glucose in order to make one copy of protein. And so this then explains why bacteria are very interested in conserving energy. All right, and so now, so that's the reason that the gene expression is highly regulated, because they want to conserve energy. They only make genes that are needed when they are needed. So that's the energy part. And then the other is that bacteria have to carry all the genes, but they're necessary. But often they don't have the needs for it. So that's also another reason for regulating gene expression. So for example, E. coli, E. coli can...
Can you repeat the second reason?
The second reason is all the bacteria carry lots of genes, and but they only need certain genes when the situation is right. So let me give you an example. E. coli, and how often do you think E. coli be able to drink milk?
Only when lactose is present? And
Yes,
to break it
and only so think about E. coli in your gut, right? Some of you drink milk, some of you don't, and I haven't drank milk for a while, and so my E. coli would just don't need genes for making to use lactose, right? And so now you understand my E. coli just shut the lactose utilization genes off, but someone else can keep it on when they have lactose comes in. Okay, so there's lots of reason to regulate the gene expression. So now we can come back to where the gene expression can be regulated. What are the regulation points? So here, so if you look at gene A as an example, the gene A is fully expressed, meaning it has is transcribed, translated to make enzyme A, and the enzyme then can carry out the enzymatic reaction. So if you take lactose as an example, is that when lactose shows up, I need LacZ or beta galactosidase to make to cut the lactose into galactose and glucose so that we can use them. But if you look at gene B, the gene B, if we want to if we don't need a gene B, C, and D, then what you can do is you can either block at the enzymatic level, so you have enzyme, or you can block translation, or you can block transcription, right? So this is an example of gene A is at the right. So gene A is fully transcribed, fully expressed, but these three represent B, C, D represent different points of regulation. And before we go into the detail, and if the bacteria has three choices, or if you are facing three choices, how do you determine which will be the mechanism you use to block the gene expression? Do you choose to block enzymatic level, or do you choose to block transcription? How do you choose? Bacteria makes its own choice, but if I throw the question to you, how do you decide which mechanism that you want to use to regulate this particular gene? If you have a gene, do you choose the approach for gene D, C, or B?
Can you repeat the question?
Okay. The question is, if you have a gene that you want to regulate, and then you have three choices. Do you use the mechanism for gene B, gene C, or gene D? Because they are regulating at three different stages, right? So if I ask you to regulate a gene of your choice, so how would you regulate? Right? You have three choices. How do you make the choice?
Wouldn't it depend on what needs to be regulated?
Yes, well, we know it's need to be regulated, but if you have to choose between three different mechanisms, how would you choose? Is it based on something, right? Based on the color, based on something?
I saw like for example, with like the lactose, next to the promoter there's like a repressor. So when the lactose is present, it binds to the repressor, and that's how it stops.
Yeah, but that's what we're talking about, the mechanism. But first of all, what's the standard you choose, right? And so let's say if I'm asking you to come to you're sitting at home right now, right? So I'm going to ask you to come to Lehman today, now, and which transportation you will, which way you will, how would you get to Lehman, like now? And well, then you say, okay, what do you mean? How long? Then which transportation method depends on what's the end requirement, right? Do you say, all right, you can come anytime. You have the entire day to come. Then you say, if I have the entire day, what do I do? Do I choose the cheapest way, or do I choose the easiest way, or do I choose? Now, how do you choose, right? So you have different criteria to choose. So I'm asking, what's the criteria for bacteria to choose which mechanism to regulate? If you don't know, the standard, I'm sorry, Ashley, I cut you off. Go ahead.
I was gonna say, I would choose if we're talking about like I guess the method and what probably would be more efficient. I would say gene D because then you don't do any work. You know, you stop it before anything else happens, so that way you're not wasting energy doing stuff that's unnecessary.
Well, based on the Lucille suggests that we should think about considering energy costs, and so because throughout the I spend a lot of time trying to convince you this is an expensive thing, and so if we were to stop at B, so if we let's look at the energy cost.
I said D,
So if
sorry,
we
like the D.
oh D, I'm sorry, D, you meant D.
Mm-hmm.
Okay. Yeah. So the option D is the has the spend the lowest energy because it didn't spend any ATP, right? Nothing happened. Well, okay, so that's the most energy efficient approach. But what about so is that the best approach? Let me give you a scenario. If you have two bacteria side by side, and one decide to make the genes or express the gene for lactose, and the other decided not to express the gene for lactose. Now, lactose show up randomly, right? And it depends on you because you don't tell your bacteria, I'm drinking milk in 10 minutes, get ready, right? Well, you cannot. And so the bacteria in your gut just sits there and wait, right? So now you drink milk and the glucose, lactose goes in. Now, which bacteria will get to grab the lactose?
The one that has the lactose.
Yeah, and so the one that has the lactose gene expressed or make the investment, and that one get to use the lactose. Now, the one that you don't have the lactose utilization gene, then that gets left out because by the time you produce, it takes time, right? And by the time you make the lactose gene, and the lactose is gone because the other guys are all taken. Do you see? So it's a balancing act. So I'm trying to open your mind to say, okay, yes, energy is the most efficient, important criteria, but if you are super energy saver, you may lose out on opportunities as well. So that's what they call the opportunity costs, right? So if you don't have that, well, you lost that opportunity. So now it sounds like, okay, this regulation is more complicated. It depends probably on how often your substrates show up, right? And so I don't drink, I haven't drank milk for a long time. So the bacteria in my E. coli, it's probably more preferable to be using the blocking the transcription. But if someone drinks lactose every single day, and well, the bacteria probably better off regulating at the gene B using the enzymatic regulation, right? And so that's an extreme example, but so now there are two things you have to think about. Okay, yes.
You said that someone that drinks a lot of milk would use enzyme B as in boy, but someone who doesn't drink it might use gene D as in dog instead.
Yes.
Okay.
So now what simplified is by gene D or gene B, but in the future, what you want to say is regulating transcription are regulating enzymatic level. So if for bacteria that like my bacteria, since I don't drink milk, it's better off regulating transcription. If someone drinks milk all the time, then maybe they are better off regulating the enzymatic level. Is that clear?
Yeah.
Okay. All right. And so this discussion is important because when you face choices, you have the bacteria, you have to understand why do bacteria make those choices. Okay, so now we're going to take a look at real life examples. So if we were to think about regulating enzymatic level, and these are the mechanism. There are two different mechanisms. One is you can have a substrate, you can have an inhibitor that compete with the substrate, and or you can change the protein or the enzyme structure, and that regulates that can regulate the enzymatic activity as well. Okay. So the first example is this. So this is a protein. This is an enzyme, right? And say this is an enzyme, and so this is the substrate. This is active substrate side. Then the substrate goes in and then cuts off. You can easily imagine that if you have, so typically people use the analogy of this is the key, this is the lock. This key goes in. Let's see what I mean by this is the key, right? And the key goes in to the lock. Now you can always, if someone is being bad, then you can stuck a broken key in there, and then that broken key can prevent a good key from going in to the lock, right? And so if you take a substrate that's only this big, right? If you take this part, if you have a substrate looks like this, that can go in here, then you can block, and then this becomes an inhibitor. Okay, so part of partial protein or anything that resembles a substrate is an active inhibitor. Okay,
So is it the red thing that's inhibiting it?
No. Hold it there. So there are different types of inhibitors. Okay, so there's a direct substrate competition inhibitor, and that's what we're talking about here. So I'm circulating the blue, take a part of the substrate, resembles the substrate. That's an inhibitor. That's one inhibitor. But you can also have what's called the allosteric inhibitor or effector, and that can go into a different part of the enzyme that fits in here, and because it fits in there, and that actually changes the active binding site and then prevents the substrate from going in. Okay, so this diagram shows you two ways of inhibiting. I'm using this to show two inhibition.
So they're both inhibiting the enzyme?
Hold on. Inhibit one, and hold on 1 second. And this is inhibit two. Okay. So any any enzyme can have multiple inhibitors. Is it clear?
Yeah, I have a question for that. The key that you said that's the yellow thing, right? The
Yeah.
substrate. So is so that's the key and the substrate. So the key is the substrate, or
The key is the substrate. A
and so,
broken key is an inhibitor. I mean, also, I'm using this as analogy, right?
yeah, and then the for the allosteric one where it binds to the allosteric site, is that something similar to like a key as well, or is that like
Yes, that's a key as well, except that key is more important. It's but you have to think the lock. If the lock is flexible, then if you jam, think about if a lock is a foam, it's a sponge, and then if you jammed another uh, big substrate, the inhibitor 2 is a big substrate, and you fit that in, and then that changes the lock, changes the lock itself, so that the key, the original key, doesn't fit in anymore, and that's a second way of inhibiting.
Okay, so I just want to make sure. So both of them can't go in at the same time because when one goes in, it changes that so the other one can't go in.
Yes, and
Okay.
so think okay. Um, I'm gonna talk too much. Yeah. Do you guys have had biochemistry? Or just get a sense of,
I don't know, not
no one had? Okay. Okay. So this will be a very, very important concept there. So the key and the lock is a typical way of thinking this. And so but if you think the enzyme is a foam, a sponge, a flexible sponge, and if you were to put this in, maybe I'll take a different way to say, okay, this is a jigsaw puzzle, right? If you think about a jigsaw puzzle, and this is made of foam, and this supposed to go in here, fits in perfectly, right? But if you have a broken piece that goes in here first, now the second one will not get in. So that's one way of preventing this uh, enzyme from acting on the substrate. Another way is that you can have a bigger component, and this is a big component. It wouldn't fit in. It has to jam in. It has to jam in. But when it jams in, because this is jammed in, and um, this side changes, becomes smaller. So you see, this is big, and this becomes small. And because it's small, and this key doesn't fit in anymore. All right. And so now you have two ways of preventing the enzyme from picking up the substrate. One is you have a broken substrate here, or two, you have an allosteric effector that's changing the structure of the enzyme. Is that clear? This is a very important concept because the protein is actually flexible. Think protein is a foam, a sponge-like substrate. Any questions? Okay. So that's one. That's the inhibition, and uh, and the next is real life examples of the enzyme activity can be changed by modification. So we have glutamine inside, so we need glutamine, right? It's one of the essential enzyme, amino acids. And the glutamine concentration can vary from, here is low, right? And so here is low, and this is high. Can't see it. All right, and then the concentration, obviously, if you have high concentration of glutamine, do you still want to make more glutamine? No, right? And so you want to make, right, so the glutamine concentration needs to be maintained at a certain level. And in this case, this enzyme can be modified by 12 different AMP, okay? And each time you need an ATP, it converts into AMP, right? And so what happens is that in real life, what will happen is that the glutamine synthase activity, so here you see the blue line here. The blue line here represents the concentration, and the orange line represents the enzyme activity. So the higher the concentration of glutamine, well, the lower the enzymatic activity, right? Because that makes sense. If you have lots of glutamine, well, you don't need the enzyme activity, right? The enzyme should be not active. And so this is an example of why why gene regulation, this enzyme needs to be regulated at the enzymatic level, this protein, right? Because having this enzyme that can be regulated quickly, then you can maintain. So the happy medium probably is here, right? Then how do you regulate? Well, that's by AMP, adding AMP group. So here, if you add, here you have 0 AMP group. That's ATP, and then that's kinase, and then puts the AMP onto the protein, right? So if there's no, if you have this is at enzymatic level, right? And if you have very or no glutamine, right? And then the enzyme is at 100% with no modification. But it can add AMP, add, probably add it to here, and then it's happy medium. 6 is about right? So in reality, what's probably going to happen is if the glutamine concentration reduce, and you're going to add more. So if it comes back down here, and then you add more. And if it's too much, and then you remove. And so that's one example.
Is AMP the only regulator for, like, does it only regulate glutamine? Or does
Yeah,
it?
so no, it's the backwards. It's this enzyme. This particular enzyme is regulated by the numbers of AMP group on the enzyme. This is enzyme very enzymatic specific. Other enzymes are regulated by other mechanism.
Okay.
Okay, so this is an example of how enzymes being regulated. And then in terms of regulation and how do you regulate, then we have what's called the feedback inhibition. And what is a feedback inhibition? So typically in biological process, a substrate needs multiple enzymes to make because biomolecule is complicated, right? It needs multiple enzymes to synthesize an end product. So this is a schematic diagram for a substrate or end product needs 4 different enzymes to make. And so if your end product is getting a lot, and then you obviously need to shut down. But if you were to shut down by a feedback inhibition, well, then you will need the end product, because if you shut down this entire process, the energy will be wasted to make enzyme B, C, and D. And so you're going to shut this whole thing down. So the in real life example is here is if you have glutamic acid can be used to make arginine or proline. And then what happens is it's more sophisticated, because they actually, well, we actually need to, if you have too much arginine, you will only need to shut down this reaction. You don't want to shut down that, right? Because you don't want to shut down proline. You want to shut down only the arginine. Okay, so the feedback inhibition is in a branched biosynthesis pathway. Then you inhibit the branch point. Well, there are other examples of feedback inhibition. Is that, well, this is complicated because they share a common process, and so each of them will inhibit one particular process. The end and the net result is this gets reduced, and so this tyrosine is reduced, then phenylalanine and tryptophan, all 3 end products gets reduced, which means these 3 are linked. One of them is overproduced, then all 3 gets reduced. Okay, so this is just to tell you, or there are different mechanisms of feedback inhibition. So at this point, up to this point, what we have discussed is regulating the enzymatic level, and it's very complex and it's enzyme specific. So there are different, you have to look at different enzymes and to understand what mechanism they're using to regulate. But usually it makes more sense if its enzyme is regulated, if an enzyme is regulated at the enzymatic level, that typically means the substrate shows up very frequently, or they need to be maintained at a particular concentration. Okay, so now we're going to talk about regulation at the transcription, at the translation and transcriptional level. Before I go in and comparing this regulation and the translation or transcription, if you block at the translation, and this is wasted. And so there's much more common to regulate at the transcriptional level. So then if you block this, then there's no mRNA is made, and so no waste. Okay, so between regulating and translation and transcription, it's much more energy efficient to regulate at the transcriptional level, and bacteria actually have a lot more mechanism to regulate at transcriptional level. Okay, so so this is really to
that last part that you said, that it's better to regulate at you said transcription level or translation?
It's better at the transcriptional level. I know it's sometimes it's if you ever confused, there are rules about thinking about this is the energy. And okay, so
and at the
the
level, that's if the substrate is not produced, like if it's not frequently needed to be maintained or regulated.
I'm sorry, I missed the first part of your question. I'm sorry.
So for the transcription level, this is when like the substrate doesn't show up often.
Yes, yes, yes. Okay. So at the transcriptional level regulation, so these are these are examples we'll be talking about. So we'll first talk about the lac operon, then we'll talk about arginine operon, and we'll talk about the mechanism of activation. Okay, so before we go into the detail, I just wanted to remind you again what are the switches or the points that you can regulate, right? So think about regulating the light in your room, right? You can't just, you have to have a switch. So you have to know where the switch is, right? So you have, even think about your house, right? So you have switch in each of the room, but you also have a main switch for the entire apartment, right? And you shut everything down. So if you think about the switch for transcription, so the promoter is your switching side of switch. And if you think about translation switch, and this is the ribosome binding site is another transcription translation switch. You can control that as well. And then you have transcriptional termination, and you have translation stop. And so I know these are all complex, and you absolutely have to keep them straight. And most common mistake when we ask for what's the start of the transcription, people will say, oh, transcription regulates by star codon. That's not right. That's the wrong answer. So I just wanted to make sure you understand all of that. Okay, so we're going to need to take a look at a real life example of the lac operon. Okay, there are some things in biology, in molecular biology, in genetics that you will benefit from having a real life example. And if you have to, if you only remember one operon, and this is it, the lactose operon. Okay, how important is this? Well, the biochemistry, the microbiologists will tell you about this. Biochemistry will talk about this. Genetics will talk about this. Molecular biologists will talk about this. And so it's show up everywhere. Okay, so you need to be able to get a piece of blank paper, be able to draw out this slide. Is that clear? Everyone needs to be able to draw this slide on a piece of white paper. Okay, so first you look at the lac operon. And so an operon, so first of all, the concept of operon. What is an operon? Operon is a unit, contains the structure gene and the regulatory region. Structure gene is the gene that encodes an end product. Typically, they are protein. Structure genes are the genes encodes a product, and typically those products are protein. And so in this case, the structure genes are these, LAC-ZYA. The LAC-Z is beta-galactosidase, is the enzyme. The LAC-Y is the permease. Okay, those are the two most important genes. And then you have the LAC promoter. Now the LAC promoter we already talked about is the DNA RNA polymerase binding site, In this case, RNA polymerase includes the core RNA polymerase and the sigma factor. That's all drilled in one. Then in the front, and this part is the key, the LAC operator. And the LAC operator is the binding site for the LAC repressor. The LAC operator and is an inhibitor binding site or repressor binding site. You see, it binds to it. And you may ask, why don't they just call it LAC repressor binding site? Why do you have to say LAC operator? Well, it's for historical reasons. The lactose operon was the first enzyme, the first gene and mutations that microbiologist was able to isolate. And so historically, it was named operator, and that's the name it's being used continuously. Okay, so so you have repressor, and that keeps it off, right? So in this case, the repressor is binding to the operator and physically blocked RNA polymerase from moving forward or from transcribing. So now the LAC-ZYA is not transcribed, so the transcription is blocked. So when transcription is blocked, if this is an E. coli, and do you think, are there lactose around?
No, because
Why?
it's not able to be produced since it's being repressed.
It is because lactose is around. When they have lactose, they use the repressor. So they don't need to synthesize anything because they have it already.
Excellent. Mamadou, that's excellent. So lactose, that's the guess, right? So there's no lactose around, so it's all repressed because you don't need the lactose. Okay, so but when you have lactose and you do need to turn it on, and what will happen is then you will have an inducer. Inducer, what happens when the inducer comes? Well, the repressor, look at these two arms. The arms changed. The configuration changed. And so now you see it doesn't fit into the LAC operator anymore. Because if it go down, it's only one of them can fit, not both. Think about me holding on to a monkey bar. If my hands goes out, and when I jump on, only one of them grabs onto the monkey bar and the other is not, right? Because the monkey bar is about this big. If I do this and I won't grab it, and me with one hand, I won't last too long. I'll fall off. And if the repressor falls off, then the RNA polymerase goes out, and then the transcription starts. Okay, so when you have lactose around, there will be an inducer to turn it on. So if you were to design an inducer to change the conformation of a repressor, what molecule you will use what molecule would you use as an inducer if you were to design this?
Allosteric effector.
Okay, that's so you allosteric effector. Anyone else? What is the allosteric effector, right? That's a question. What molecule would you use as an inducer? Say it again.
Would it be lactose in this case?
Yes. Lactose is used by the bacteria as an inducer. All right. So I hope you are smart. I'm sorry. I hope you are impressed how smart E. coli is. Your friend E. coli is, right? Or as smart as we are. We decide the lactose will be the best inducer, and that's exactly what E. coli is doing. When you have lactose comes in, and the lactose binds to the LAC repressor, changes the conformation, and voila, you have the genes expressed. Okay. So what happens in nature is this. So if this is a graph uh, looking at the relative increase, and so you have bacteria is growing, the cell numbers are growing, and the total protein, so here's total protein, right? And that's increasing. And then if you add lactose, well, when you don't add lactose, this lactose, the beta galactosidase, you actually have to remember, is the product of LAC-Z. Okay, there's no way around it. You just have to remember. You just have to remember beta galactosidase LAC-Z. LAC-Z encodes beta galactosidase. I'm sorry. Hopefully, you repeat enough time, you will memorize it. Okay, so you add lactose, you see beta the LAC-Z increase. To make things even more, I wanted to impress you even more about E. coli. And what happens is that if you, this is looking at, the red looks at the bacteria, numbers of bacteria. And the blue looks at the beta galactosidase. And if you give the bacteria a little bit of glucose and lots of lactose, what will happen is the bacteria will grow happily, and they will use only glucose. And then only when glucose used up, here, look at this. The E. coli will actually stop and think for a second, not think for a second. They actually have to look and look around to say, can I find glucose? And they say, well, no, I can't find glucose. There's no more glucose left. Are you sure? Yes. Okay, no glucose left. Now I'm going to have to use lactose. And then they turn out the lactose. Okay? It's almost like if you like orange juice and you go open the refrigerator, there's no, well, if you have orange juice, you drink all the orange juice. But you like the Tropicana. And then when that thing, you finish all of it, and say, okay, all right, now I'm going to choose. I have to drink some other juice, right? Because I have. And in the end, it's like, okay, I'm going to drink water, right? And so the point here is that bacteria actually able to differentiate the two sources of carbon, glucose and lactose, and they will actually use glucose first, and they will actively inhibit the enzyme for lactose, because lactose is there, but they cannot be expressed because having glucose allows the bacteria actually inhibits the lactose. All right, and so this is a plug advertisement for my advanced microbiology class. I won't teach it in the spring, but they for different reasons. But just to tell you how complex the E. coli actually is. And there's a question. Yes, go ahead with the question.
Is it because glucose is just easier, or is it just like at any time there's a switch?
No, it's just the glucose. It's easier, and it's the most common source of carbon, and they actually have a mechanism to inhibit the glucose. I mean the lactose utilization, because it's more efficient to use glucose, because you don't need to make excess of. You don't have to express LAC-Z.
Okay, thank you.
Okay. All right, and this is so now back to here. Okay, so hopefully you are absolutely impressed by E. coli and this mechanism. Okay, so you absolutely have to know this operon and this mechanism. Okay, now I wanted to add a little bit more about this part of the induction part. Okay, so so this lac repressor is actually formed by two copies of the same protein. And so you actually have two copies and they each bind to a binding site. So it's an inverted repeat. Okay. And it's in the major groove, right? And we talked about it. And so it's a dimer. And so that's why if it's a very specific fit and having a lactose here changes the conformation, and so one doesn't fit anymore, and so that's why only one of them will fit and will fall off. And this is a very common process. Okay, so this protein, typically the DNA binding protein, typically have a recognition helix, and that fits into the major groove. And that structure is stabilized by a stabilizing helix. And so they typically have helix-to-helix conformation or the structure, and so for this interaction. And this stabilization can also happen with ions as well. So you can have a protein rely examples, right? These are unique examples. You can have a zinc ion and stabilize a recognition helix, and this then fits into the protein domain, I mean the DNA. And this is a very common structure that you have two proteins, two copies of protein, identical protein, and they have two copies. Now you get them together. This is dimer and dimerized. And so now you have two recognition helix, and this is what the lac repressor is. Okay, so let me back to here, and now you have this two copies of the lac repressor and binds to it. And so if you think about back here, now you fully understand why lactose binding will change the conformation of this dimer. This is actually two proteins and dimer, and then the binding no longer fits into the lac operator. And so then the binding is not as stable, it falls off. Okay. So now I wanted to give you a different example of arginine. And so earlier we said arginine biosynthesis can be shut down if you have too much arginine. And so you can actually demonstrate this in actual experiment. And so if you look at this graph, and you have cell numbers, and you have total proteins are measured, right? And so the bacteria grow. That's what you and when they grow, they will need to make arginine constantly. So the enzyme, well, you need arginine, right? So the enzyme of arginine biosynthesis enzyme, this is the blue line, and that's being made all the time. Well, you need arginine, so it's right, they're making it so that they can make arginine. But at this particular time point, if you dump a huge amount of arginine to the bacteria, now arginine or any amino acids are really good, a bacteria just take them in immediately. All right. So arginine goes in, and arginine biosynthesis enzyme gets shut down immediately. Why?
Is it like the threshold or something? It's that it doesn't do anything for it anymore, so it stops.
Well, because it's a feedback inhibition. Remember, because if you have too much arginine, why do you need to spend energy to
Energy.
transcribe and translate to make the arginine biosynthesis genes? and this actually tells you is a good example of telling you that the proteins in the bacterial proteins are actually need to be constantly made because they recycles pretty fast. The half-life of bacterial protein is pretty short. So they are reacting quickly. So what happens at the molecular level? Well, how do they actually react, you wonder? Okay, so this is the arginine biosynthesis operon. Arg operon, so you have Arg C, B, and H. Those are the structure genes, and you have an Arg operator. And you have Arg promoter in RNA polymerase. And you have a repressor. And repressor here is not binding to it, and the transcription is going on. However, there are cases when you do want to shut it down. I can't ask you the question anymore because it's written. Maybe I should block it next time. When you have arginine, right? What happens when you have arginine? When you have arginine, all the gene biosynthesis gene needs to be shut down. How does it achieve that? Well, arginine actually is the co-repressor, and it changes the conformation so that the repressor normally doesn't bind. Now it can fit perfectly. Now you shut down the transcription. So the transcription is blocked. And this is a real life good example of how bacteria can actually have a molecular mechanism to do the feedback inhibition. So earlier we said the feedback inhibition can rely on the allosteric protein or effector can change the conformation of the enzyme. But that's changing the conformation enzyme. What if you want to shut down the production of a whole bunch of enzyme to shut down a pathway, right? In this case is a biosynthesis pathway. The arginine need to shut down this pathway. Well, then you need to shut down the transcription of the genes, and this is then how it's achieved by changing the conformation of a repressor, making it an active repressor. So in this case, this is an inactive repressor, and this is an active repressor complex.
Is the inactive repressor when the transcription is proceeding, and the active is when?
Yes.
I also wanted to ask, why is it like this? Is it just the way that it is, or like compared to the other?
So that's a good question, right? And so why is this? Well, one way to think about this is E. coli has probably existed on the earth much longer than we do, right? So they evolved to have this the perfect system that allows them to react. And one concept perhaps here I can introduce is the wild type. And so the concept of bacteria have wild type is that they exist in nature, and they do because that that they encounter real life, and they are most efficient in doing that.
I have a question. So the repressive is always there. It's a matter of enabling it.
Correct. Yes.
Okay.
Yeah, in this particular case. So the way you think about this is the different genes, and there are different examples of mechanisms of regulation, right? And so this is an example of having the repressor there. And so then the repressor can be shut down. I mean, this transcription can be shut down immediately. Next, I'll give you a different example of a different mechanism. Oh, yes, go ahead with the question.
I think, the one you did, the last one, it is the same as the glucose and the lactose. So in the presence of, if we have both of them, like when the bacteria is using this repressor for the lactose, in the absence of the lactose. So when we have the lactose and we have the glucose at the same time, the glucose will be like a co-repressor, right?
Okay, wait, hold on. Um, so Mamadou is asking the question when have glucose and lactose around, and there is actually, well, you would guess these mechanisms are a lot more complex, right? There's called metabolic inhibition, which I won't have time to discuss. I think your textbook actually does have a description about explaining how glucose it actually inhibits. But we don't have time to discuss. And if you're interested, you can read more. But for this particular class, all we need to understand is the lactose, how lactose actually physically can regulate the lac operon. And I'm not asking you to understand how glucose can inhibit. I'm not asking you to know glucose inhibiting lactose expression, because there's other mechanism involved. It's more complex than we have time for. Good? Okay, so now I'm going to give you a different example of regulating gene expression. So in this case, maltose is an example. So maltose is a sugar that doesn't show up that often. So naturally, we don't need to express the maltose utilization gene. And in this case, so then the maltose has a promoter. It doesn't bind to the RNA polymerase that efficiently. So in this case, it's a weak binding between RNA polymerase and maltose promoter. So the RNA polymerase goes up and falls down quickly. Okay, and so goes up, falls down, and doesn't stay, and so there's no transcription happening. And there is a maltose activator protein. And that maltose activator protein requires an inducer. And then the maltose activator protein will bind to the binding site just upstream of the promoter and then start the transcription. So the 2 points I want to make, or questions. What molecule should be the inducer? Anyone want to guess? What molecule would you use as an inducer to turn on the maltose promoter?
Maltose, right?
Yes, maltose, yes. All right, you see, E. coli is as smart as Mamadou, right? As smart as us. And so, and then the next question is, why is the activator binding site upstream of the promoter. Remember, where is the repressor binding site? It's here, right? The repressor binding site is here. The activator binding site is there. What we call the upstream. Let me type it down. Upstream, downstream. Okay, so no, let me. How do I delete this? Okay. This is downstream, and this is upstream. Okay, so we use these two terms to describe the upstream and downstream, and so to describe the relative position, we use the stream as an analogy of the direction of the transcription. Okay, and because water in the stream only goes in one direction, right? From the upstream to downstream, usually from higher to lower. And so if you have, so it is important that for you to notice the binding site is upstream of the promoter, so that this can stabilize the promoter and then start the transcription, right? Because you don't want your activator to bind here. Now, if it binds there, then it physically blocks the transcription. That wouldn't work, right? So this is called the activation. And so the activator can be directly upstream, but there are activators can be far, far further upstream of the promoter. And in that case, the DNA actually bends over so that the activator can come back and then stabilize this interaction. So that's another example of activator. So now what we talked about, the lac operon, the arginine operon, the maltose operon, and there's one particular transcriptional regulation that is very interesting and very specific. It's only discovered in the last 20 years. And so here is what's the two components. First of all, what are the two components? When we think about two-component regulatory system, there are two components, right? One, this sensor kinase is one, and this repressor is the other one. Okay, so that's two components. That's component one and component two. And what's unique about this is that each component have two different states. So component one is a sensor kinase, and it sits across the membrane. It is capable of binding to the environmental signal. And once it binds to the signal, it becomes a active kinase. And then it puts a phosphate onto itself. So now this has a phosphate, and it becomes a kinase. An kinase is an enzyme that can put a phosphate onto a second protein. So now this kinase is now putting the phosphate onto the response regulator. So now the phosphate is putting onto here, and this response regulator becomes phosphorylated. And phosphorylated response regulator is capable of binding to operator. So this here, an operator, so this binds to the operator, then it will block the transcription, right? Structure gene, promoter, and block the transcription. So now what the net effect is that the environmental signal is actually regulating gene expression directly. Or another way to think about this is the bacteria is able to respond to environmental signal in regulating its gene, right? And so this is shutting off. So if you, meaning that the bacteria sees this molecule turn off and then stop the transcription of a particular gene. And you can have other examples of which we didn't draw here, is that you can have a signal molecule and this phosphorylated regulator then binds to upstream of a promoter, then that will turn on the transcription of genes. Then that, in that sense, the signal will be able to turn on bacteria gene expression. Okay, so altogether, the bacteria can rely on the two-component regulatory system to turn on or to turn off a gene. So, right, and so typically a bacteria will have somewhere between 20 to 50 two-component regulatory systems, 20 to 50. Why? Because each sensor is evolved to sense a particular signal molecule. And these are very specific structure matching. And so if they match, and then they turn it on. And if either turn on or turn off. So you can have a bacteria capable of sensing 20 or 50 different environmental signals or environmental molecules and controlling gene expression. Okay. Any questions on this? Okay. The application of this two-component regulatory system, here's an example. So we said the bacteria can sense attractants and repellents, and so if a bacteria is swimming towards repellent, if you are walking towards something smells terrible, what would you do? You keep going?
No.
No, you stop, right? So a terrible smell will stop you from going, and you stop, right? If you smell gas and say, okay, that's bad, I'm going to stop, I'm going to turn around. Now, what happens to bacteria? If they are swimming towards a bad thing, would they need to stop, right? Okay, so, well, wait, let's see. How would bacteria do? Well, they have a sensor on the membrane, and then the sensor will phosphorylate, activates the key A, and the sensor goes there, and then once they sense bad things, they become active, and then they will phosphorylate key A. Then the key A will phosphorylate key Y. You see, key A has this phosphate, and then react with key Y, and the phosphate goes to key Y. And the key Y is a protein that can jam the flagella rotating machine. So you were rotating, right, and running towards, we call it counterclockwise, right, rotation, and the bacteria swimming happily. But then all of a sudden they smell things bad, and that bad smell triggers key A phosphorylation, then key Y phosphorylation, and key Y goes to the promoter and jams the promoter. I'm not the promoter, I'm sorry, rotor. And now the flagella rotating direction reverses. What happens when they reverse? They're going to tumble, right? They go from swimming, yeah, to tumble. Tumble, they just stop in the original direction, right? So they stop from going towards. And what's very, very interesting is that the bacteria also have a key Z, a protein. A key Z is a phosphatase. Phosphatase is the enzyme that removes the phosphate from any protein. So now you have key key Y that is dephosphorylated. Why do you do that? Well, and that's because you need to be able to sense another bad smell, right? Because you start tumbling, and soon you're gonna start swimming again after 5 seconds, right? And you're gonna swim again. But when you swim again, you may run into repellent again. So now you're ready to sense the next repellent. Okay, so this is an application of how bacteria actually uses two-component regulatory system to sense the outside, and actually in this case is not regulating gene expression, but physically changing the behavior of a bacteria. So in today's lecture, let me quickly summarize. We focus on the regulation of gene expression. We first spent a lot of time talking about why I'm trying to convince you why bacteria have to regulate its gene expression. It's all energy. It's all energy, right? We calculated how much ATP or how many ATP you will need to make one copy of bacteria protein, or one typical copy of bacteria protein. And right, so now we know it needs to be regulated. So then we think about how you regulate the gene expression. You can regulate the enzyme activity, or you can regulate the enzyme synthesis. And majority of the mechanism that we talked about is at the transcriptional level. And main reason is, well, because transcription is the most energy efficient. If you think about energy synthesis, I mean the enzyme synthesis, you have transcription and translation. If you want to shut it down, it's much cheaper to stop the transcription. And so here are the three mechanisms. We are using an example. And lastly, we talked about a two-component regulatory system that allows the bacteria to sense the outside environment and then regulates gene expression and actually physical behavior. And so I'll leave you with this overview of the example overview of the regulation and at different points. And one key thing is that it's a balanced approach. Where do you block, or how do you regulate? Do you regulate enzymatic activity, or you regulate at the transcription? And they each have pros and cons. Regulating at enzymatic activity, because the enzyme is there, that allows you to react really fast. But regulate at gene expression, and that allows you to maximize saving energy. But you may lose out at the competition when you all of a sudden start drinking a milk, right? It's like if I go into the fridge, grab a cup of milk and drink, and my E. coli will be shocked and say, wait, I haven't seen milk for ages. I don't have any, right? enzyme, and so now my E. coli get lost in the competition for lactose. Okay, so I will
I have a
stop here
question.
and yes, I will stop here and answer any questions that you have.
Um, well, I don't know if this is like pertaining to microbiology. I was just curious, like, so if you haven't drank milk in a long time and you just drink it, is it harder or does it take a longer time for them to break it down because they have to like remember or something, or is it just fine?
No, the E. coli reacts in the same speed for lactose. I'm just making it dramatic, right? And but
Okay, I'm just
for
curious.
E. coli always have the well, E. coli always regulates lactose using at the transcriptional level. And so when you drink milk, it always takes a transcription and translation, then making it and then using it. And obviously, there are a lot more interesting mechanism behind, but that's the level of detail we want to know at this point. Okay, I'm going to stop the recording.